What am I doing? I do not remember. Hi, if you are joining me, welcome. Let's go on. Shut this window a little bit. And welcome if you are joining me. Hi guys, welcome if you are joining me. If you have any questions, please feel free to ask away. I am just downloading my group tuition session, so I have a bit of time. And so therefore I am here to help and answer any questions you might have on A-level biology. So if you are watching, then please feel free to ask a question. I do not need that anymore. Hi guys, welcome. Welcome, welcome, welcome. Is there any topic area you would like me to cover, or I could just start talking? That also works. Oh. I had this thing today. I went to fill up the car and I unfortunately decided to pick the petrol pump where there was a leak at the, it was like the connecting part to the pump, and it meant that I got diesel on my hands and the smell was just awful. And I can still smell it now, even though I've washed my hands like a million times. It has not gone away. It has been very frustrating. But hi guys, if you are joining me, if you have any questions, please do feel free to ask away. I am waiting for the downloads, so I thought I would pop on. Oh, do you know what I'm going to do? I'm going to show you. I did a thing for my group tuition session tonight, and we were talking about the manipulating genomes. Please could you go through DNA replication? George, of course I can. I will in 1 second, because I really want to show my really cool diagram that I did today. This one. I'm going to show you a little animation. You are very welcome. You are very welcome. What part of it that you're struggling with? I'm assuming that you are potentially year 12, but you might be year 13 and just trying to do a bit of a recap. And if that's the case, that's completely fine. That's good that you're picking up. But this is my diagram that I have that I wanted to show. So if you're not with me on my other channel, I'm actually displaying my board. But what I was super excited about is we were talking about restriction endonucleases, and I was talking about how the fact that you can have different restriction endonucleases and they can cut DNA up, and that will then be separated on gel electrophoresis kits. And what you can then do is see the different fragment sizes that you can get and how they would separate if you have all the different ones. So I was like super sad but excited that I could show this. So George, you are year 12. You just started it in year 12. You don't understand how the enzymes work with unwinding and unzipping. Right, okay, that's an interesting one. So when we look at DNA, So if we're talking about the structure of DNA, you know that DNA is a double helix. So I'm just going to do very basic diagram. So you know that DNA is a double helix. Hi. And because you know that DNA is double helix, you also know very basic. Oh, that's a really dodgy line that I just drew there. Never mind. You also hopefully know that DNA is anti-parallel. So when we look at the strands and we're talking about our DNA polynucleotides, we're talking about the fact that they are anti-parallel. And so when we say they're anti-parallel, we're talking about the position of the 5th carbon and the 3rd carbon. So I'm just finding, give me 1 second whilst I pull over. Here's one I prepared earlier for you, because that'll make it much easier. So we're just looking at DNA replication at the moment. And I will bring in a little bit of diagrams. Hi, I have AS exams in May. When should I aim to finish all the bio content? If your AS exam is in May, give me 1 second because I actually know the date of, I want to say it's quite early in May. It's like the 12th. So if I were you aiming to finish those exams, I would say you want to ideally have your content done. I put it in here. Yeah, AS paper 2, AS paper 1, 11th of May, and then the 21st of May for your exam. So I would say you ideally want to have finished that content by the beginning of April. So you've got a month to just go over exam paper after paper after paper. You should hopefully be doing practice questions as you go, but I would say ideally you want to finish the content by the beginning of April for those exams coming up in, well, your first one's the 11th of May for biology. Right, back to what I was just about to do for George. George, I haven't forgotten about you. Don't you worry. I am coming back. Okay, so here's one I prepared earlier. So if we're looking at the double helix, we're looking at the fact that we have this anti-parallel strand. Give it a second whilst it saves what it needs to do. What you would then note is, she says, you can actually come on here. You're going to play. Oh, why is that not working? It's not, come on. It's definitely. Oh, there we go. It's because it's got an animation on it. I've forgotten my other slide's got an animation. There we go. So the anti-parallel strand. Ah, perfect. I'm glad you're here, George. Perfect. Right. So anti-parallel strand. So we've got the double helix. We've got this anti-parallel strand. And what you have between your nitrogenous bases here is you have these hydrogen bonds. So you've got the fact that you have this twisting that happens as you go every, thank you. These are my diagrams I made myself. It's a funny one because some of my PowerPoints that I quickly grab the stuff from in these sessions, I'm like, these are my old stuff. And they're like, oh, where's my new stuff? But this is a diagram I am proud of. It took a long time for me to do this. There's another one actually I had up. Where did I have it up? See, I'm going a little diversion. I will come back, I promise you. There it is. This was one I was super proud of, but I've not used it in a long time actually. Let's see if I can get it on here. This is one that I did on my Illustrator as well. Are you live on YouTube? Yes, I am live on YouTube as well. Hi Callum, good to see you. So yeah, so when it comes to DNA, when it comes to double helix, we know that it's twisted, and so therefore we have to remember these key parts. Now, if we're talking about eukaryotic organisms as well, we need to actually bring in the fact that it's not just that it is a double helix. It's not just that it is, let me just make this a bit smaller. got antiparallel strands. You've also got the fact that it is associated with histone proteins. So we have to take all these bits of information into consideration. So the first thing, depending on what your teacher has taught you, they have potentially tried not to confuse you, and only talked about DNA helicase. But they may, hi guys, welcome. For those of you who are on TikTok and want to, I am on YouTube as well, so I'm kind of trying to do both. People don't seem to ask questions on YouTube, or it doesn't work as well on the live chat compared to on the TikTok, so I kind of use you both. I'll ask for your advice when I'm asking illustrations for any program I make in the future. Ah, perfect. Absolutely. Please do. I love what I've learned in the skills department. Obviously, I'm not the best. I'm still learning, and I'm learning it all by watching YouTube videos, then trying it out myself, and then coming back. But yeah, I love it. I absolutely do love it. Going back to this, DNA helicase. So George, I've not forgotten, sorry. DNA helicase is needed for two things. The first thing is about relieving the strain. Now, technically, it's not DNA helicase, but we allow it in OCR. So there is another enzyme which is called gyrase. So your teacher may have taught it to you, they may not, but with OCR, they only really focus on DNA helicase. Then you have DNA polymerase. And you have ligase as well as the other one that they want you to know. So DNA helicase is going to relieve the strain. Technically it's gyrase, but we're going to say it's helicase. DNA helicase is going to break the hydrogen bonds. They did mention gyrase, and this is why I do mention it. So I make sure I will give you information that is what you need for the exam, but sometimes having a bit more helps. And so it really depends on your teacher as to whether they want to go a step further in the specification or not. But the ones you need to name are DNA helicase, DNA polymerase. Ligase technically isn't named until you get to the module 6 manipulating genomes, and that's when they throw in the ligase, but I always bring it in here. But if they're asking it at an AS level, they will actually give you that bit of information and then say, what are the other enzymes and what do they do, for example. So DNA helicase is going to break those hydrogen bonds. So if we are going to separate our strands, this is my strand and I want to separate them. So right now I can't separate that diagram itself, but if I just get the actual picture up, 1 second, so that I can do this. If I do DNA structure, let's go this one. Right, I'm going to change the color. A second. And then I'm going to bring the other one in. So I have my strands here. Oh, crumbs, no, I don't want that. Got to be very careful what I'm copying here, because I want it to be just the bits that I need and no more. And I'm hoping this will work. There we go. Right. So you can tell that I'm live because I'm literally going to make little bits that work and then maybe not. Okay, so here I have my double helix and I have my anti-parallel stranding. I can tell it's anti-parallel because of the position of the carbon. So I have very basic and a nucleotide. I have my deoxyribose sugar, because we're talking about DNA, or ribose sugar if I was talking about RNA. So then I have my 5 carbons, carbon 1, carbon 2, carbon 3, carbon 4, carbon 5. So that's the first thing to note. We're talking about DNA, so at carbon number 2, we know there's a hydrogen and not a deoxy ribose. It is a let's try that again. It's a deoxyribose and not a ribose because it has not got an oxygen at carbon number 2. If I was looking at it as being a ribose sugar, then I would have the same structure. The only difference would be, if I just move this over here, I'm just going to quickly change that. Hopefully this will work. She says, there we go. Instead of it carbon number 2 having the hydrogen, it's going to have the hydroxyl group. So if it's a ribose RNA nucleotide, it's got a ribose sugar. If it's a DNA sugar, it is DNA deoxyribose. I was literally like, it wasn't a pause on this, by the way. It was me pausing in my brain. Just, you know, ask any biology questions. Love it. Why? Why should you? Yeah, why should you? Why bother? Okay, let's go back up. There we go. If you are looking for the functional human gas exchange components, please go check out my transport in animals, no it's not, my exchange surfaces videos, because it's all there. But yeah, lots of practice means that you are going to figure out what you don't know by asking the questions. You can then fill those gaps that will help you. Tony, I just saw Tony. How are you? Anyway, back to this. So if I'm looking at my antiparallel strand, I have got the nucleotide with the 5' end up at this part and the 3' end at the other side. And obviously if I look on the other strand, I can see on the other strand I have got my 3' end and my 5' end there. Harry, you're absolutely right. You could ask ChatGPT to do it, 100%. Why not? Could you go over plant hormones? My year 13 mock is tomorrow. Oh, Anna, if I've got time, I will. But I am answering George's question first. Okay, so what do we need to do? We need to take this double-stranded helix and we need to separate it. So what we're doing is we're going to break the hydrogen bonds. Now, if we were to leave this and not build a new strand, these would just come back together. And because they would come back together, they would form hydrogen bonds again. So we need to ensure that they are separated in a way that means that we can build a new strand. And we can build that new strand, I'm going to do very basic here, from one nucleotide to the next. That was an awful nucleotide structure. I do not know what happened to that one there. That went really dodgy. So what we need to do is we need to break the bonds and then allow free nucleotides to form complementary base pairing coming together, and then we need to build the phosphodiester bonds between the two, and then we keep doing that as we move down the strand. But because we are doing it and because we can only build from a 5 to 3 direction, it means that the RNA polymerase can only move in one way. So what we have to understand then is, not that one, this one. One second. Here you go. So DNA polymerase is only going to be able to form these phosphodiester bonds between the adjacent nucleotides, and it can only build in a 5 to 3 direction. So if this is my template strand and there's my 5' end and there's my 3' end, what will actually happen is it will build in a 5 to 3 direction. And because it's building in a 5 to 3 direction, it's going to move in opposite directions. And that will result in the new strand being built. And the new strand, as it's being built, is being built one nucleotide at a time. And so therefore, what we get is, at the moment, our DNA polymerase has built a very small section. But what's going to happen is this one at the top is going to continue. And as it continues to build the strand. And so we get what's known as one strand, which is the leading strand, and one strand that is the lagging strand. So we can see that that one continued, whereas this one is going to detach. Oh no, it didn't. Did it not detach? Have I removed that animation? Oh no, it did. And so you get, there we go, let's try again. There we go, a detaching, where it has built the strand, and it built the strand in a 5 to 3 direction. And when it detaches again, it is then going to end up at this end. So we've got a fragment here and a fragment here, whereas this is now up at that end. And it keeps going and keeps going. Does that make sense? And so what you end up with is, with the DNA replication side of things, is you end up, let's just go on this one, where because the helicase is going to make its way up through the linear chromosome, and the DNA polymerase can only ever build in a 5 to 3 direction, what that results in is these little fragments where the ligase has to come in and finish joining the fragments together. So the ligase is the last bit to just connect those fragments by building those phosphodiester bonds. Does that help? Has that explained that part to you, George? If you are still with us. You are very welcome. You are very welcome. Right, Anna, you want some plant hormones. I think, hold on. My PowerPoint is literally pausing on me now. I've got too many things open with high resolution pictures. What did you learn as DNA polymerase, Anna? Be careful on that one. So your teacher might not have mentioned the ligase yet if you haven't done your manipulating genome, although I think if they haven't taught that to you yet, they're doing you a disservice, because then they're just going to throw in a new one. So ligase, no, ligase is for connecting the Okazaki fragments. Ruben, if you want to go to my channel, you want to look at 5.1.4 or 5.1.5. I've actually got it in playlist for you, and it says it's neuronal communication. So it's got all my videos on the nervous system. You've only just started the manipulating genomes, so you haven't done genetic engineering yet. Well, that's a bit... It's a bit rough. They're only teaching you the DNA ligase now, but yeah, DNA ligase is needed for building these Okazaki fragments. But a lot of the time what they do is they give you a question where they tell you the ligase, if they were doing it as an AS question, and you are expected to do the other two. Yes, it is A-level, Ruben. That is exactly what it is. It's aimed at the OCR. specifications. So if you're AQA, you need to just double check the connecting parts to your exam board. But coming back to hold on, I'm going to show you. So whenever you are doing your content and revision on the content, the one thing I would say you should always do is, no, there we go, look at what the specification says for you. So semi-conservative DNA replication to include the role of, as I said before, if you're just revising what the spec says, the enzymes helicase, DNA polymerase, and the importance of conserving that genetic information. So they're the only two for the Module 2 content, Year 12, that you need to know. But when you get up to the manipulating genome bit, that's when you need the extra bits about the ligase. But it is important that you are aware of what it does. So I try and show you where it all interlinks. Okay, Anna, you said you wanted to look at plant hormones. So let's go for, what would we name as the hormones? So if we're looking at plant hormones, the first thing I would say is let's just do a quick list of the hormones. So what are the hormones? We've got auxin, we've got gibberellin, we have got abscisic acid, ABA, we have got ethine, we have got, so auxin, for those of you who, if you are here doing AQA, that is your IAA, by the way. Gibberellin, ABA, ethine. What am I missing? Am I missing any? Why can I not think off the top of my head now? I can tell it's getting late. My brain's going, not so much. Right. Let's start there and let's add some in as we go and see if we can work out what I've missed, if I've missed any. So auxin, what do we need to know about auxin? Well, auxin has the fact that it does apical dominance. What does that mean? When you look at the root tip or the shoot tip, you should know that you have got your apical meristem. So the apical meristem is at the tip of the root, it's at the tip of the shoot, and therefore when we talk about apical dominance, what we're saying is that auxin is going to cause cell elongation, and that cell elongation is going to cause the cell to get bigger. And so we want that to occur at the tips of the root, the shoot, the apical meristem, or it can happen in the cambium, which is the part, if we were looking at the xylem and phloem, so this is my phloem and that's my xylem, the bit in between is the cambium, which is also made up of meristem, and that's going to therefore push out and divide by mitosis and then differentiate into what is required, whether it's phloem or whether it's xylem. So the apical dominance is where that will actually prevent lateral buds. So if these are my lateral meristem, they are inhibited from growing because the apical dominance is that we want the plant to grow upwards towards the light, because they're going to be competing against other plants for light. So they want to get high, but they have to control how quickly they grow upright, because obviously they can't move around and there's the force of gravity. They can't grow too tall too quickly because wind and gravity will just cause them to collapse and fall down, and they won't have exposure to light. So they do it in a very controlled manner. So apical dominance is where there is a high concentration of auxin, and that high concentration of auxin will promote the growth of the apical cells, but it will prevent, so it will inhibit the lateral growth. So it inhibits the lateral growth, but it promotes the apical growth, and that is known as apical dominance, and that's because of auxin. Now, cell elongation that's going to occur, this one, and we're just writing this very basic notes, cell elongation is where the auxin will bind to a receptor and it will cause hydrogen ions to be pumped into the cellular wall. So into the cell wall, that is going to activate expansins, which are enzymes which are going to break the crosslinks, and when it breaks the crosslinks, I went over how it works for the essay, but I forgot. I do remember. Yeah, you do only do IAA and AQA. That's right, for your plant response. So the cell elongation part of it is the bit that you do. Do I do A-level biology videos for EdXLB specification? No, I don't, but the majority of the content is the same because I tutored EdXLBs last year. So go and double check the video specific to the content and it should link quite nicely. Your content is less to know, but your questions are harder, I would say, on the application front for EdXLB. So my videos are OCR A, and the OCR A is very specific to the EdXLB content and also very connected to AQA. So cell elongation, hydrogen ions are going to be pumped into the cell walls. That's going to activate expansins, going to break the crosslinks. So therefore the cell wall is now flexible. So if the cell wall is flexible, that means the cell can take in more water by osmosis. And if the cell can take in more water by osmosis, what we get is, that's my original cell. Because it's taken in more water, it's going to get bigger. It is going to elongate. And so by making the cell wall flexible, that allows for more water to move into the cell by osmosis, because the cell wall actually is preventing the cell from taking on too much water, and that's what creates the turgidity, which means that the cell is able to withstand the pressure and it stops it from bursting, because you have got the wall, which is a layer, a thick layer, which is going to prevent that cell from bursting when the water is trying to get in. So it allows water to get into a point, and then it stops any more water going in unless it has been signaled by the hormone, which is a plant hormone, to elongate. And that is what happens when it comes to the elongation. Now, if we're looking at tropisms, then what that would then mean is when I have my shoot tip and the light is coming from above, let me move the ethene because that's going to make it confusing. When I've got the light coming from above, the cells at the tip are going to detect the direction of the light. They are going to therefore produce auxin. That auxin is then going to diffuse down, causing these cells to elongate. As it diffuses down, causes those cells to elongate, the shoot grows up. Now, if I have the light coming from one direction, what's going to happen is we're going to end up with the auxin being produced by the tip and it diffusing down more down the shaded side. And so therefore the elongation that occurs at the shaded side is more than the elongation at the light side. And what you want to happen is basically if I'm a plant and you are the sun, if I bend because I have the chemicals being sent down to the cells on the shaded side, causing more cell elongation on this side, I'm now going to bend, and therefore I'm going to expose both sides to the sunlight. So I'm maximizing the rate of photosynthesis because now both of my leaves are being able to access the light. And that's what it's about. It's about that maximizing the absorption of light for photosynthesis to occur. More photosynthesis, more light absorption, more electron transport chain, more ATP production, more light dependent reaction, therefore means more light independent reaction, and that results in more of the organic compounds being built. And so that the plant is able to then use them and utilize them to grow, whether that be through the carbon compounds being used in respiration, whether that being building the cellulose, which is needed when the mitotic division is occurring and this nucleus is being divided. You are going to have to build more cell walls as the cytokinesis is happening and those cells are splitting. So we need those organic compounds, and that is where the light independent reaction produces the triose phosphate, and that triose phosphate can then be used for any of the necessary molecules. And if we need the storage, because it's a plant, it will then store that as starch. So it really depends where they want to go. But looking at the hormones, auxin, the auxin is causing the cell elongation. The cell elongation means that the cell can take in more water. It can elongate, which causes the bending towards the light. How do we know any of this? Well, we know this because we've had scientists in the past do these experiments. You had Darwin, you had Wentz, you have so many different scientists who've done these investigations for you. Now what we're doing is we're helping you to understand it so you can then come up with the new experiments so that we can utilize it more. We can use it in a way of maybe synthesizing a fake hormone for commercial use, because as the population is growing, as there's more and more humans, we need to feed them. So if we need to feed them, can we utilize the chemicals that we've learned about, find a faster or better way? Because we're talking about a hormone that's going to bind to a receptor which is then going to activate things inside the cells. So if you understand that all of this is linked to proteins, to biological membranes, to enzymes, if you've got a solid foundation of your Module 2 content, all of biology is about putting it into context. So they can throw any novel idea at you if you understand the fundamental parts. So the auxin and the apical dominance is about, if I have a high concentration, it's going to promote growth, but it's going to inhibit growth elsewhere. And if I've got the right concentration, I can, and that's the optimum concentration, I can actually utilize the growth, have the right yield that I want, but minimize the cost that it may be to ensure the production of it. So it's this whole thing of how can we manipulate, use science, use our knowledge to manipulate the world around us to make it do things at a faster rate for less of an effect on, let's say, the environment, etc. Things like that. So I don't know where I was going, but anyway, that's where what would be your advice on approaching and revising application questions for OCR? Millie, the best thing I could say to you is practice, 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 as many practice questions as you can. Do you think you could briefly go, probably not briefly go over the nitrogen cycle, but I do have a video on the nitrogen cycle. Can you help with serial deletion test and the type of question about that? Ria, the serial deletion question could come anywhere they want with that type of question. The reason being is it can come into so many things. Actually, I'll tell you about it with auxin because it links quite nicely to a question on auxin. Paper 1 today, and there was a question on how exercise increases breathing rate, and I wanted to talk about accelerator nerve, but for AQA that's paper 2 and 3. Right, okay, so I'll come back to that about the AQA and that in 1 second. Linking it to serial dilution, if I wanted to do an investigation looking at auxin and how auxin affects the growth of the apical meristem and the lateral meristem, I would want to use specific concentrations of auxin. So therefore, how would I actually make those concentrations? Well, how I would make those concentrations is I would be provided with potentially a stock solution, and that stock solution is going to have, let's say, 100 parts per million of auxin concentration. But then what I'd want to do is I'd want to do a tenfold serial dilution. So this is where they could link the serial dilution question to the content or the question that you're doing. So serial dilution can come in so many different ways. As long as you understand how to do the serial dilution, that would be what I would say they could ask on this one. Because we know apical dominance is to do with the concentration, we therefore need to be able to control it. If we're going to do any type of investigation, so for example, gibberellins, just to lead in this, and I'm not forgetting about the others, gibberellin is about stem elongation. So again, we could do a question looking at stem elongation and looking at how the different concentrations of gibberellin have an effect on the growth of the stem between the internodes. So when you look at the branches that come out, so if I do my next branch coming out down here, so there's my lateral buds. This is what's known as the internode. So that section there is the internode. So whenever we're looking at the stem elongation, we would be looking at that region there and what's the distance between it. So it could be a question that's saying, okay, what's your independent variable? My independent variable is my concentration of the hormone auxin, the concentration of the hormone gibberellin, whichever exam question they go down the route of. How would you prepare that? And it won't be as simple as saying, how would you prepare that? But you should be thinking, right, my IV is my independent variable. I'm going to change the concentration of the hormone. How am I going to do that? Because I should be giving them 5 values for my independent variable. So that would be 5 different concentrations. So if I'm going to do a tenfold, I could do 100 parts per million, 10 parts per million, 1 parts per million, 10 to the minus 1, 10 to the minus 2, and so forth, all the way down to 10 to the minus 11. I don't know, wherever you want to go on that one. So as long as you understand how to take the content that you have and put it to the question in front of you, that's where they will be asking the question. So that's merely what I would say when it comes to application questions. What do you know content-wise, and then how can you apply it to the specific example that they have given you at whatever question they've given you? So gibberellins is about stem elongation, but gibberellin is also about seed germination. So do you understand that seeds are going to be where there is stored energy, and that stored energy is in the form of starch? Well, that starch needs to be broken down, but we're just getting out of winter. It's really cold outside. It's actually not that cold anymore, but it was a lot colder. And so therefore, there was no point in seeds germinating because there's not very much light. It's quite cold. So the kinetic energy, because of the thermal energy, is going to be low. Therefore, the chemical reactions are going to be low. So do you understand that your enzyme module 2 comes in to seed germination? It's not just understanding which hormones do what, it's how they're interconnected. Gibberellins are the hormones that are going to cause the seed to germinate. The seed will only germinate if the gibberellin has been activated, because the seed has the right conditions. There is the oxygen, the temperature, and the water concentration. So therefore, I may do an investigation where I look at different water concentrations and which is the right water concentration to cause the seed to germinate at a faster rate. And so when I was teaching the IB, my students had to actually do and plan these investigations for themselves. So they would do like a preliminary where they would do a little test, and then they would then actually do the investigation. So what you'll get in your exam for OCR is you'll be given, students have done this, how could you improve their investigation? Or you need to now plan it. So you should be saying IV, DV, CV, I need to do repeats, I need to calculate my mean and standard deviation, and I need to do a statistical test. So that's what you should always be doing when it comes to any type of investigation on the different plant hormones. Now, let me, before I answer your question, I will come back to that, but let me just quickly finish. So ABA is to do with water stress, and it's to do with your guard cells. So it would close the stomata, and it closes the stomata because it is going to prevent transpiration because there is water stress. ABA is also needed for the production of what's known as antifreeze. So we've just gone through a very cold period within winter, so therefore we need to have our seeds able to still have chemical reactions occurring because of the cold temperatures. There's going to be less kinetic energy. If the cells themselves were to freeze, if their freezing point was maintained at the level that it normally is, what would happen is the water, because of water's property and those hydrogen bonds, it would actually expand out, and that would then break and damage the cells. So the cell will produce a chemical, an antifreeze, which is going to prevent the cell from freezing by lowering the freezing point. Also, it actually does seed dormancy. So the hormones actually work antagonistically against each other. And the reason being is because it's all about the correct environmental signals. It's not this hormone on its own, this hormone on its own. It's all these different hormones at different levels maintaining the plant's ability to keep growing. And then the last one, just to finish the different ones, is ethene. Ethene is about the fruit ripening, and also about the, what else is it about? My brain is totally switched off right now. Wow, you can tell I'm getting really tired. This is so late at night, and I just did an hour teaching. Negative feedback, not always necessarily negative feedback. Negative feedback is where there's a stimulus and you are going to reverse the effects of the stimulus. So you need to be careful that you're not just throwing in the information that's not relevant to the question. So yes, there could be a form of negative feedback, but you need to make sure that it's specific to the topic area and not just throw it in there as an answer, because that's what some students do, especially AQA students in the 25 marker. They will throw in things like negative feedback, and it's not relevant on the question. And so because it's not relevant, of course, leaf fall, I was right. In my brain, I was like, am I thinking about leaf fall? But I am thinking about leaf fall and fruit dropping. I'm getting tired now. It's not going so well. Leaf dropping. Right, you had a thing. There was a question about atrial fibrillation, and the company claimed their watch can detect atrial fibrillation, but they only use one. who is young and only for one date. So, I mean, that's an easy one, isn't it? If you've got only one person, your sample size is so small. If you've done it only one day, that day could be so stressful for them. Do you know what happened? Do you know? And also their age is going to have an effect. So I have a thing that my friend has this on their watch. It's really interesting. So they have the watch that does the same thing, and it detects when they are in a stressful situation, and so it sends a message to say, you need to have some quiet down activity because you seem to be getting quite stressed. Your heart rate has elevated. And that's like when you're surrounded by the kids. Interesting. Or when the noise level gets too high. So you have to be aware of all the extra things. You can't just claim that this does this and that it works amazingly and it tells that there's a problem with the heart. It's really interesting about the bits that they won't tell you, and therefore you should be thinking about that and how the environment has an effect. Right, I'm going to do... Oh, Harry, I love the fact that you're on here trying to be the person who is writing negative comments, and what you're not realizing is that what you're doing is actually making my life seem so much better because it makes them actually push me out more. So thank you. I appreciate that. So yeah, abscation is the word that I should have used. You're absolutely right, and not lethal. I've just been lazy. So I completely agree that ChatGPT and OpenAI should be available. The one thing I would say to you is just be cautious that you are not writing stuff that's incorrect science. This has just come off my For You page again. I really... Oh, thank you. That's really lovely to see. I'm sorry I haven't seen it until now. Right, I am going to say good night to you all. Thank you for joining me, and I'll be back on Wednesday as I always do. Tonight's just if I'm waiting for something to download, I will pop on if I have the time and I'm not too tired. But yeah, hi Zara, how are you? I do remember you. How's it going? Although this is the bad thing. People jump on here and then I'm like, oh, hi, I'll keep talking for another 25 minutes. Bye. Have a good evening, guys. Thank you for joining me. But yeah, I need to actually go because I need my bed. I need to sleep. I had a busy weekend this weekend. I was in London. You are very welcome, and I hope that has helped. If you're still struggling before the mock tomorrow, obviously you've got my playlist specifically on the topic area that drops it into different small bite-sized videos on each of the different ones. Amazing. That's really good. Well done. Right, I need to go to bed. So good night, everyone. Thank you, and have a good evening.